Ladies and gentlemen, I'm pleased to introduce the 2020 Shaw Prize laureates in life science and medicine, Gero Miesenbach, Peter Hagemann, and Georg Nagel. Luigi Galvani, in the late 18th century, was the first to appreciate that nerve cells could use electrical signals to pass information. Fast forward to the 1930s, when a brain surgeon, Wilder Penfield, seeking to understand the origins of epilepsy, probed regions of the brain with an electrode to find that the stimulation of specific areas elicited reproducible motor activities shared by all of his patients. Thus, the origins of a brain map. In more recent years, electrical stimulation of very specific regions of the brain has been used for therapeutic treatments, such as in the control of motor dysfunction in Parkinson's disease. Given the complexity of the human brain with around 86 billion nerve cells, each of which may make as many as 10,000 connections, totaling over 1 trillion or 10 to the 12th contacts, the task of a complete understanding seemed a distant prospect. And yet now, through the combined studies of our 2020 Shaw laureates, Gero Miesenbach, Peter Hagemann, and Georg Nagel, we have a new optical tool and an approach using directed gene manipulation to map the circuits of virtually any neural network. Miesenbach was the first to appreciate the power of an optical probe to elicit an electrical response from targeted nerve cells. His team used the visual pigment rhodopsin, responsible for converting light into electrical signals transported to our brain, to engineer an artificial signal into nerve cells growing in the laboratory. His lab's particular application was to use the fruit fly rhodopsin, responsible in the fly for detection of light with as little as one single photon, able to elicit a controlled response in a single cell. They showed that a fruit fly rhodopsin introduced into a vertebrate nerve cell incubated in the laboratory could be stimulated by light, and further, that the use of this fruit fly rhodopsin could be used with the application of light to control a neurocircuit in the fly itself. The origins of the current revolution in optogenetic control of neural networks can be traced to the introduction of his 2002 paper, where Miesenbach concludes that, quote, "Since sensitivity to light is built into each target neuron, advanced knowledge of the spatial coordinates is unnecessary. Large numbers of neurons can be addressed simultaneously and precisely without undesirable crosstalk to neighboring cells that are functionally distinct." Now, rhodopsin from more evolved multicellular organisms, such as us, requires a set of other protein molecules and a small time lag to elicit a signal in response to light. For applications in neural network mapping, a faster and simpler light-responsive protein was sought. Years before the Miesenbach invention, and working quite independently, Peter Hagemann found a speedy photoresponsive channel rhodopsin, a class of rhodopsins that is also an ion channel and differs from visual rhodopsin, which acts only indirectly to open an ion channel. This channel rhodopsin orients a photosynthetic microorganism called Chlamydomonas to swim toward a source of light. Shortly thereafter, and again working independently, Georg Nagel found that microbial rhodopsins could be transferred by cloning into vertebrate cells where light would directly elicit a flow of ions. Here now was another key breakthrough in the development of the optogenetic tool. A single protein with a built-in light sensor triggering the opening of a current-generating channel. In 2002 and 2003, Nagel and Hagemann published two critical reports of channel rhodopsins 1 and 2, the second of which, found by Nagel, elicited an extremely fast light-induced increase in ion entry into cells to generate the kind of membrane depolarization needed to excite neurons. Within 5 years, the idea and specific application of channel rhodopsin 2 led numerous groups around the world to create ever more powerful approaches to the optical control of neural circuits. In an understated retrospective review, Hageman and Nagel expressed some mild surprise that, quote, "We did not expect the research on molecular mechanisms of algal phototaxis or archaeal light-driven ion transport to interest readers of a medical journal when we conceived and performed our experiments a decade ago." But as we've seen repeatedly in these Shaw Award ceremonies, the power of basic science to reveal the rules of nature and their application to the human condition is perhaps the greatest invention of mankind. Thank you.